My research is in forestry species. We work developing trees that are better adapted to a changing environment, trees that are more resistant to pests and pathogens, and trees that grow more. We have essentially three main species that we work on. One of them is pine. We also work in poplar, and we also work in eucalyptus. The research that I do is particularly focused on trying to identify genes that allow us to identify those trees that are the best suited to be used in forest plantation. A lot of the work that is done in the lab is what we call computational work, and that essentially involves the analysis of DNA sequence that is derived from these individuals that we study. So for example, these are poplar plants that have been genetically modified with a gene that we are characterizing. We believe that this gene essentially regulates the size and number of vessels that the stem of this tree would have. What we think is that it creates additional vessels, larger vessels, and we are interested in that property because it is believed that that property can make potentially a tree grow more. But what you're seeing there are trees that are modified for this gene. What you see in the greenhouse are essentially populations of the species poplar as well as pine that we study. Essentially what we do in these individuals is that we plant them in the field, we characterize their DNA, and we characterize also their properties for growth, wood quality, disease resistance, among other traits. And then based on their DNA sequence and based on those traits, we identify the genetic markers, the DNA that is involved in the control of those traits. So one of the significant concerns that we have is with climate change. We know that the climate is changing. We know that temperatures are getting warmer. That can have very dramatic consequences in our forests, both in natural forests and also in planted forests. Our intent is really to identify what makes one individual perhaps better adapted to the environment, a range of different environments, compared to perhaps an individual that has to grow under very strict, limited conditions. The idea is that if we identify the genes that regulate these kinds of properties, we'll be better prepared to address challenges that will come from climate change, challenges that will come also from the introduction of new pests and pathogens which happen every day.